So my case study will be focusing on AML, acute myeloid leukemia. This is just a snapshot of what my lab is currently doing at Nottingham Trent University. We are focusing on understanding the immunological ecosystem of AML using a variety of different approaches, all of them developed at Nanostring Technologies, and these include immune gene expression profiling, novel 3D biology technology, which looks simultaneously at RNA, protein, and DNA, and of course, last but not least, digital spatial profiling to get insights into the immune architecture of this relatively rare but very serious condition. So this is my background slide. Chemotherapy is still a standard of care for most patients with AML, in spite of recent approvals of novel agents, at least in the US. Primary refractory or resistant AML, as defined by not achieving complete remission, is still a significant issue, occurring in approximately 10 to 40% of the overall patient population. And unfortunately, the 5-year overall survival from first relapse of AML is only 20%. So this is clearly largely unsatisfactory. This is an area of unmet medical need. We need to identify new molecularly targeted and immunomodulating agents. This is a high priority for those in the field. So again, there is an urgent need to develop predictive biomarkers to help identify patients who are more likely to respond to cancer immunotherapy. And we know that investigations in the solid tumor field have indeed identified predictors of response to immunotherapy. And I'm just mentioning here the interferon gamma-related RNA profiles and also tumor mutational burden. So this slide summarizes our preliminary work in this area just by showing that the immune gene signatures can really stratify patients into distinct clusters. This is shown in panel B. This is unsupervised hierarchical clustering of immune cell type specific scores. These were calculated using the pan-cancer immune profiling panel, and again, the Encounter platform. So this slide shows quite clearly that we can use this information to separate patients into an immune-infiltrated cluster and into an immune-depleted cluster. And is this clinically important? Yes, it is, as the Kaplan-Meier curves clearly show here. So patients with immune-infiltrated bone marrows had a significantly worse clinical outcome, both in terms of relapse-free survival and also overall survival time. This is apparently a counterintuitive finding, but the most likely explanation for this could be that inflammation and also interferon gamma-induced negative checkpoint may really make patients patients' prognosis worse, so they can dampen anti-AML immunological responses. So the research questions behind our AML pilot study. So having previously shown that immune profiles can assist outcome prediction in AML, can we use DSP to gain insights into the CD3 neighborhood of immune-infiltrated and immune-depleted AML? We all know that having good neighbors is important. So what about the CD3 neighborhood within the bone marrow biopsies? And can we use DSP to identify immunological biomarkers of clinical outcome in AML? So we were privileged to be able to run samples in Seattle through the Technology Access Program. So we just used 10 archived bone marrow biopsies, FFPEs, from patients with newly diagnosed AML. There is no need for us to go through the clinical details. I just want to point your attention to the fact that we just collected more than 7,400 data points from 10 bone marrow biopsies. This is an incredible amount of information. So we were able to identify 24 regions of interest. At that time, the immuno-oncology panel just encompassed 31 invisible proteins. I know, Joe, you can do much more than this. So this is going to be the next step. Yeah. So at that time, we generated more than 7,000 data points. So we used 3 visible targets, CD1, 2, 3. I mentioned this marker quite a lot previously to identify AML blasts, and then, of course, CD3 to identify T cells, because that was the research question in our mind. This is a summary of the analytical strategy. The composition of this panel has already been discussed. So we decided to analyze samples on a per-sample basis, defining T-cell infiltrated, above median across samples, and then T-cell depleted AML cases, but also on a per-region of interest basis, defining CD3 high and CD3 low regions of interest within each individual bone marrow biopsy. This is a snapshot of the region of interest capture in these 10 bone marrow biopsies. So as you can see here, we used rectangles and circles to identify T-cell infiltrated areas. within the bone marrow biopsies. So this is more clearly shown here. So today is a sunny day here in Amsterdam, so I'm hoping the audience can see the green dots in there. Those green guys are CD3 T cells. So we identified areas with a brisk T cell infiltration and others which were T cell excluded. And there was a very nice correlation between the count of CD3 barcodes and our ability to categorize the regions of interest as being low or high just based on a visual inspection of these slides, which is a nice quality check for our data. This is an example of a T-cell enriched bone marrow biopsy. Again, there are areas in here with plenty of green dots being CD3 T cells which infiltrate those specific areas within the bone marrow biopsy. And this is an example of a T-cell excluded bone marrow biopsies with very few, if any, CD3 T cells. So we need to understand what the difference is between a CD3 high and a CD3 low region of interest. This is just to show you that you can really try and make correlations between T-cell infiltration patterns and the expression of other proteins. As you can see on the right-hand side of this slide, for instance, beta-2 microglobulin barcodes were significantly higher in the CD3 high regions of interest. This makes a lot of sense. You have more T cells, you have more antigen presentation going on within your tissue. And then also the key 67 barcodes were higher in CD3 low areas of interest. This probably being due to the fact that there is more tumor cell proliferation if there are not enough T cells to try and constrain tumor cell expansion. And also we detected a nice correlation between CD45-RO, so a marker of chronic T cell activation, ongoing immunological responses, and high CD3 status, and also PD-1 barcodes, which also suggests that your T cells may be dysfunctional, so they may express higher levels of markers related to T cell exhaustion. But I like speculations, scientific speculations, of course, not financial speculations. So it's very nice to also see T-cell duos. So if you go really deep and look at T-cell to T-cell interactions in tissues, this is lovely to me. So this is a very nice picture showing that those T-cells are talking to their neighbors. So there is probably an exchange of information going on within the tumor microenvironment. But it's also very important to see whether protein expression correlates with RNA expression. So we also analyzed those samples using the pan-cancer immune profiling panel, and we found a very nice correlation between the CD3 high versus low status and also RNA scores, which is shown in panel C more specifically. So higher CD8 RNA scores, exhausted CD8 RNA scores, and T-cell scores, and also cytotoxicity scores in the CD3 high samples as determined by DSP, so by visual inspection. And this is a more detailed snapshot to show that we detected significant and positive correlations for most of those markers when looking at protein data and also RNA data. So there is a lot of information we can really garner from these experiments, and to understand why the RNAs are translated into proteins or are not translated into proteins, which I guess is going to be crucially important to understand the biology of this tumor. So then we built correlation matrix, looking at protein profiles in CD3 high versus CD3 low bone marrows. So this tells you that the CD3 neighborhood is really completely different in more heavily T-cell infiltrated bone marrow biopsies compared to the T-cell depleted bone marrow biopsies, with the CD3 neighbors mostly being B7H3, so a negative checkpoint, PD-1, B cells, monocytes, beta-2 microglobulin, and beta-catenin. And this is completely different to the CD3 neighborhood in the T-cell low bone marrow biopsies with just CD45-RO, again, a marker of chronically activated T cells, and also PD-1, a marker of exhausted CD8 T cells. So there is a huge difference in terms of CD3 neighborhood in the T-cell infiltrated versus the T-cell depleted bone marrow biopsies. So this is a very small patient cohort, and we had response data just in 9 out of those 10 patients. But it's very tempting for clinicians to speculate about correlations with clinical outcomes. So this is what I did in here. And this slide shows you that you have higher PD-L1 barcodes and higher expression of VISTA, which is, again, a very novel negative checkpoint in patients who failed to respond to chemotherapy. and also higher levels of proliferation within the bone marrow, Ki67, and higher levels of BCL2 and also FOXP3, a marker for Treg cells, in patients who failed to respond to chemotherapy. So this is an indication that you can really collect a lot of meaningful information just from a small number of samples, and then also try to correlate this with clinical outcomes. This slide just shows you a protein-protein similarity matrix. So this is looking at the big picture. So we identified 4 potential protein signatures, shown here with numbers 1, 2, 3, and 4. So in the interest of time, I will not focus on 1, 2, and 4, but I will just focus on protein signature number 3. So the list of proteins within that signature is shown in blue. So we then needed a greater number of patients, of course, to try and correlate that potential protein signature with clinical outcomes. So we used TCGA AML data. So this is a publicly available collection of data. So these 7 genes, were abnormal in approximately 1/3 of sequenced patients with AML, and this is shown here. So we have abnormalities of those proteins in a considerable number of patients with AML. And then we also used fancy online tools to try and understand the biology behind those proteins. So as you can see here, there is a number of gene ontology biological processes which are significantly enriched in correlation with those 7 proteins, and also KEGG pathways. And then this was a particularly interesting finding for me. There was a clear correlation between abnormalities in those proteins and TP53 mutations. So TP53 mutation is a clearly adverse prognosticator in AML. So those protein profiles were significantly correlated with TP53 mutations, and also with a hot immunological microenvironment within the bone marrow, and this is shown in the heat map in panel A. And also panel B shows significant correlation with patient outcomes. So patients with abnormalities in those proteins had a significantly worse clinical outcome. And this was associated with a hot, so more heavily immune-infiltrated bone marrow tumor microenvironment. So the take-home messages from my case study. So transcriptional programs that reflect higher immunological infiltration enrich in a subset of patients with AML, and they have the ability to stratify survival, which was unexpected when we first embarked on these studies. But this also means that immune-infiltrated tumors may be amenable to immune checkpoint blockade, similar to the conceptual framework in solid tumors. You may want to target those patients because they are more likely to respond to immune checkpoint blockade. And DSP may provide really useful insights into the immunobiology of AML. So we detected distinct CD3 neighborhoods in T-cell rich and T-cell poor bone marrow biopsies, and we identified predictive biomarkers, so protein profiles which capture elements of T-cell biology, PI3K, AKT signaling, but also significantly associate with mutations in tumor suppressor genes such as TP53, and last but not least, they do correlate with poor outcomes. So this is really my vision about these studies and where is this field going. So immune gene and protein signatures could really be integrated with existing prognosticators, and they may serve as therapy predictive markers for AML. They can really support the development of a kind of super signature to better stratify patients. And this is my last slide, just acknowledging collaborators in my lab, mostly Jay Badake-Kulato, who has been performing the Nanostring work, and clinical collaborators in many, many sites across the world. Great. Thank you very much.